In this video, we will discuss about the treatment or gene therapies for hemophilia. In our previous video, we have understood that hemophilia is a genetic disorder. That is, it arises because of the mutation in the DNA. So, in order to treat such a genetic disorder, we need to either correct that genetic mutation or need to find a way to produce that uh, protein in a normal form and within the body, or somehow introduce that normal protein within the body so that it can perform its function. And the best way to do this is with the help of gene therapy. So let us start in the chronological order, or the therapies that have been established at first. First one is the replacement therapy. So what happens in the replacement therapies? The clotting factor, that is the different hemophilias has different has lack of different factors, such as hemophilia V lack factor 9, hemophilia A lack factor 8. So these based on the patient, that uh, clotting factor are being transfused to the patient, and this kind of therapy works from within the this kind of therapy is in demand at a time period between 1950 to 1960. After 1960, what happens is that there is an advancement in the therapy occurs, and instead of producing this cofactor artificially, people are now preserved the plasma from the patients that does not have hemophilia, and then transfused this plasma to the patient that have hemophilia. Uh, and this therapies and this kind of this strategy will exist or will in demand was in demand from 1964 to 1980. After 1993 to present, what happens is that there is a purification. Earlier the whole plasma has been injected. Now the protein that has been deficient or has been mutated in the patient are being purified, and gene sequence of this purified protein are being identified, and with the help of different therapies that we will discuss in other therapies, this kind of gene are being transfused to the patient so that it can exist for a longer duration of time, and sometime it will edit the gene of the patient to correct the mutation. So let us understand. One of first approach is the non-viral vector. So there are two kind of approach within this non-viral vector can be followed. One is the in vivo approach, another is the ex vivo approach. In vivo approach, the plasmid is directly injected. Plasmid mutated that contain the correct form of mutated protein or mutated gene that is been directly or therapeutically delivered to the patient, so that with time the transcription and translation of this plasmid occurs, and the normal form of proteins, these factors are being exist within the patient. And the ex vivo approach is that cells from the patient are being taken out and are being cultured in the lab, and then being coat, and then these plasmid are being transfected in these cells, and these cells are then these genetically altered cells are again implanted to the patient. And so in this kind of approach, it has been found that from the previous studies that even after 300 days, the traces of correct form of factor are being found in the patients. The next one is the viral vector. So this non-viral vector approach is kind of like transient transaction, and this viral vectors approach are kind of like stable transaction. As it will, as in this case, there is a possibility of genome integration is more, as the transfection occurs with the help of sub virus vector. So in this case also two kind of approaches there. One is in vivo approach, another one is the ex vivo approach. The different in viral vector and non-viral vector is that the transfer of this vector are occur with the help of virus. It is packaged into the virus vector and then therapeutically transfer. Same is the case of in the case of ex vivo. In ex vivo also the transduction occurs. Transduction occurs of the transgene vector of interest package. Uh, Transduction occurs after of the transgene vector of interest are been packaged into the virus, and these transduced cells are then restored to the patient. So various viral factors that can be used are the adenovirus, retroviral, adeno-associated virus, lentivirus virus. Next one is the our last one is the genome editing. This is the latest approach as uh, before uh, before the CRISPR-Cas9 system, genome editing was not so popular as it is difficult to do editing at a specific site. So after CRISPR-Cas9 genome editing become popular in most of the genetic disorders. So what happens in this case is that DNA can be cut with precision at a target and in such a way that now the uh, DNA are open for repair, and this breakage of DNA can be filled with the corrected DNA sequence. So this kind of approach are can be tested, can be do in vitro or by using different delivering strategy to confirm the efficiency of this gene editing. After that, this gene editing can be when the gene editing can be successfully implemented, then it can also be done via in vivo approaches. So these are the approaches for hemophilia with the help of gene therapy. So that's all for this video. I hope you like the video. You will get some knowledge enhancement, at least at the basic level. So thank you for your time.